to present my research work for this Diamond Jubilee celebration of IIT Delhi. You all can see my screen, right, professors?
Yes, yes, we can
Okay,
see and
okay,
hear you.
okay.
Okay.
So I'm Soutik Betal. As I've been already introduced, I'm assistant professor of Department of Electrical Engineering, and I have my laboratory of nanorobotics and medical devices, where I'm performing the further works for the in vivo experiments, and hopefully this technology will come soon in the market for human testing itself. So uh, before beginning here, I want to so this presentation is about world's smallest medical robot of size 120 nanometers. Uh, so this smallest robot have applications in biomedical field particularly, and as I've been introduced, this has also got its place at Guinness World Record for the smallest medical robot. Now, this magnetoelectric nanorobot, as it is named, is basically a, a electromagnetic device which is of very minute scale, and this robot have a remote control mechanism, and it also have the switchable and invisible electrical hands, a magnetic legs, and a magnetic body. Now, these robots can, yes, I'm just going to the next screen. Yes, this robot can sense the electrical field of a cell. Basically, this electrical field is the negative electric field of a cell generated by the phospholipid bilayer. Uh, these robots can permeate in a targeted cells by the process called electroporation, and this magnetically controlled electroporation is also a phenomena which we have published with the name of magnetoelastoelectroporation. Thirdly, these robots, a group of these robots, can carry or transport the cells to unlimited distances, and also to desired locations. These have a huge implementations in neuronal path regeneration, which I will specify in the later part of my slides. Lastly, these nanorobots can carry for biomolecules and can be delivered to a particular location. For example, chemotherapeutic drug, protein molecules, or DNA molecules. Now, when we are talking about robots of such a minute size, uh, the electrons which basically perform all the electronic activities in bulk machines will not work at nanoscale because of the electron tunneling effect. The electron will not stay inside the system. So it needs a radical idea to create really such a minute device to perform the robotic activities. So to really come up with this radical idea, we have used one of the technique which is called magnetoelectricity. Let me explain. So electromagnetism have shown a huge impact in last 2 centuries actually, due to its huge applications in transformers, induction motors, and generators, like you name it, all the electrical machines. But as this electromagnetism works very well in the bulk systems, the converse effect of electromagnetism, which is magnetoelectricity, have its own impact at the minute scales. So let me explain what is a magnetoelectricity. As from the name, it can be understood that a magnetic field will generate electricity. This is perfect. This phenomena can be observed in basically heterostructures, fabricated heterostructures. I will give you an example here where you can see there is a sandwiched heterostructure with two magnetic layers and magnetostrictive layers and one piezoelectric layer, which is imposed between these two layers. When an AC magnetic field excitation stimuli is applied to this device, the two layers, the magnetic layers, will start expanding and contracting because of the magnetic dipolar reorientation following this magnetic field wave. Now this strain is passed over to the a middle layer, piezoelectric layer, and it will generate the electricity. As you can see, the voltage fluctuations generated. Interestingly, this electricity produced will the amplitude, the frequency will be completely the excitation magnetic field frequencies. So from the Heckman diagram, this magnetic field is generating the stress. The stress is further generating the polarization change in the piezoelectric material, which will transfer this pressure wave into electrical signals, and it will generate the electricity. This is the phenomena called magnetoelectricity. Now, here is the structure, which is the simulations, where you can see there is a core shell structure. The structure is a magnetostrictive cobalt ferrite layer, which is coated with barium titanate piezoelectric shell. So when an AC magnetic field fluctuation is applied to, or AC magnetic field excitation is excited to this device, the core will expand and contract, and this exerts pressure on the shell. This shell, on the other hand, will generate electrical dipoles and very localized high electric field. And as I said earlier, the electric field generated wave will follow the magnetic field excitation frequencies. Now, the key thing to work for the simulated device to work is, firstly, the the shell should be single crystalline. The reason of this shell should be single crystalline is that if the dipole is created, the electrical dipoles are created. If it is not single crystalline, they will cancel each other, and there will be no net charge or no net surface voltage generated in these nanostructures. So the basic property of this single crystalline is analyzed here by electron microscope. So this is the core cobalt ferrite. It is coated with barium titanate using Pacini method. And then using the selected area electron diffraction pattern, we can see the cobalt ferrite single crystallinity, as well as the barium titanate shell is also single crystalline in nature. This is one of the best outcome also from this project, because I will explain in the later slides also. It is very difficult to fabricate such kind of single crystalline layer, particularly in these structures. Why? My next slide will come with this analysis. But here is also another radical experiment. This experiment is very innovative, called electron holography and off-axis electron holography, where we can see this electric and magnetic, invisible electric and magnetic contribution of this core cell structure using this electron holography method, where you can see that there is the electromagnetic behavioral change from core to shell. I will explain this in the next slide. Lastly, the electron diffraction, so the EDS measurements shows that the cobalt ferrite, which is in this fabricated device, in our fabricated device, is all confined at the core, whereas the barium and titanium is spread it throughout the shell of this structure. Now here comes the electron holography method, which is a very new technique, and seldom papers have explained about this. Here, what is happening is, first, a nanostructure is exposed to the condenser lens of a STEM, scanning tunneling electron microscope, to analyze. So this is first exposed to a 2 Tesla magnetic field. Next, we create electron diffraction. You might have seen the optical diffraction pattern, which is used for all the optical microscopes right now. But electron diffraction pattern is created using a biprism. When this sample is analyzed using this electron diffraction, we can monitor the electromagnetic behavior of this nanostructure. But the another key thing, this measurement, which is also one of the toughest thing, was that to find the same nanostructure, flip flip the grid which contains this nanostructure 180-degree manually, and then again find the same nanostructure from thousand of the nanostructures on the grid and analyze it. The reason for this is that when we extract the phase from the flipped and the top surface analyzed nanostructure, the addition of this phase will give us the magnetic contribution. The subtraction of this phase will give us the electric contribution. But here you can see from this analysis that how we can visualize the north pole creation of the on the particle on this nanostructure and the invisible magnetic field lines which is in the vicinity of this structure analyzed through the electron diffractogram. Again, when the sample is flipped, you can see the south pole is can be seen in this nanostructure and also the magnetic field lines. But most important thing among of this experiment is that when the top layer was exposed to when the top surface was exposed to 2 Tesla, it created a ferromagnetic memory. And when we flip the sample, we can see that the magnetization direction, as in the figure E, is continuing even when it is flipped. The magnetization direction is keeping itself in the same direction. This is called ferromagnetic memory and nanoscale. Such kind of analysis was never shown earlier in any of the publications or any of the research. Lastly, excuse me. Lastly, when as I said that on the addition of the phase, we can see the magnetization of the core. The whole magnetization is confined at the core. And when the subtraction of the two phases give us the electrical dipole creation on the surface, you can see the structure now looks big because the electrical dipole is all contained on the surface. The magnetization is all contained in the core of this structure. This thing was again analyzed on an orthogonally placed nanostructure, and here you can see the complete envelope in figure D. You can see the complete envelope going from north to the magnetic field envelope going from north to south, and same analyzed when it is flipped again. The ferromagnetic memory here was also monitored, and the dipole creation. One another key of this fabrication technique, and another accomplishment you can say, is that no matter which technique we use to measure the surface morphology of this structure, we always have similar kind of structure, as you can see here from the atomic force microscopy image. And in black and white phase images, you can see that how the magnetic field envelope is in the vicinity of the nanoparticle and how the electric field is all confined. This electric field lines is confined on the surface of the structure. So we are showing magnetoelectricity at nanoscale. With all the invisible line forces can be visible using electron diffractogram. Again, since I said that this was all in DC magnetic field, right? But if we really need to have this electric field generation by AC magnetic field, so that there should be a fluctuating behavior on the of the shell, which was so this can be seen here from the PFM measurement, where the atomic force microscope tip at 0 voltage can show some topography of these particles. But when this tip voltage was plus 10 volt, there is a phase change on these small nanostructures. And when again we flip the electric field to minus 10 volt, we can see the switching behavior of the nanostructures, and the phase changes to positive side. The electron holography measurement was further confirmed using the MFM, the magnetic force microscopy, where you can see at one side there is a north pole creation, or where there is the deflection of the tip, and the other side where there is the attraction of the slip. So here is the whole magnetic bipole being created in this nanostructure. This on the image here, using the optoacoustic signal, we were trying to see that how far on the electromagnetic spectrum at variable frequencies, how far the nanostructure will follow this stress transfer from core to shell. And lastly, in this image, you can see the bulk ferromagnetism being created in a particular amount of the nanostructure using the magnetometer measurements. So, as I told earlier, that such kind of structure is very difficult to fabricate. The reason for this is so we are trying to put a perovskite tetragonal lattice on that, like grow a perovskite tetragonal lattice on the top of a cubic or spinel structure. The spinel structure have a dimension of 7.65 Armstrong cube, wherever the perovskite structure have dimensions of 4 into 12. So this kind of mismatch, this kind of lattice mismatch will never allow the growth of a single crystallinity, because this will always try to bring the polycrystallinity or amorphous nature in the cell fabrication. But using proper pressure application, proper heat, proper step-by-step heat application, as well as controlling the ambient environment, lead us to the to grow a 45 nanometer uh, thick single crystalline layer of tetragonal lattice on a cubic lattice substrate. Now, importance is, okay, we have fabricated whether they are really, this structure is really stable or not. To see this, we perform the GP analysis, where we can see the stress being confined at the and the interface of these two layers. And high resolution microscopy, along with GP analysis, shows us the strain is all confined at the surface of the layer rather than on the interface. This rotational strain, which can be seen in figure A here, that how this whole strain is all confined at the surface, which further increase the integrity of the structure. Okay, now the device is already created. Now, how it will function? Lastly, here we measure the size using variable techniques like Melbourne's meta-zeta-sizer, which use the dynamic light scattering method, and the electron microscopy itself to find out the sizes of these nanostructures. Now, there were two Nobel Prizes in 1991 and 2003. These both were about single ion channel study in cells. And also, the second Nobel Prize was for structural and mechanistic study of ion channel. So, what is ion channel and how it is important? Here, if you assume a cell, our human cell or any of the living cell, you will see that whenever there is an electromagnetic change or the energy change nearby the cell membrane will try to open nanopores on the surface, temporary nanopores on the surface, and uh, try to bring the equilibrium by the sodium potassium ion inflow outflow. Such kind of such kind of behavior generates the electrical charge differences, and when this energy, the electromagnetic energy change, is very high, then this group of cells will generate these electrical energy in the form of neurons and pass this information to brain or central nervous system that you please do something, remove the place from this electromagnetic change. This is called ion channels. Now, my main objective of it was to engineer of ion channels in a human cell. So such kind of techniques of engineering this nanopores was earlier been done by various stem researchers, but that was a very uncontrolled method. Firstly, what they do is they put cells in these cuvettes. They put a very high electrical charge, almost 110 kilovolts, to generate localized microvolts at centimeter scale. Now, the cells found to be near the negative electrode, they opens temporary nanopore. As you can see in this image, they open temporary nanopores. And when the electric field was removed, they again closed this nanopore. Also, there was research where they tried to open the same thing, same nanopores on single cell by using nano electrodes. This technique have a very great application in foreign DNA transfer by opening these nanopores on the cell membrane and introducing the DNA molecules, or to introduce a biomolecule inside a non-penetrating cell and cure the cell or kill the cell. But the limitation of earlier techniques was very high electric field. Bulk devices were used to apply such a minute localized electric field using such a big machines. And also the technique was very uncontrolled, because it results in death of the cells, as you can see here, how these cells start deforming and creating permanent pores, and at the last they just result in cell death. So my target was using this nano device, the device we fabricated, we will open one single nanopore on one targeted cell and will not affect any of the untargeted cell on its pathway. Now I will just proceed with this with the experimental section. Here in this slide, I've shown that how the electric field generated. What is the electric field generated when we apply a magnetic field by monitoring and calculating the magnetoelectric coefficient, which is the transformation of magnetic field to electric field. Now, the idea here was so we have this phospholipid bilayer, which is kind of a polar layer where we have the phosphate head, which is PO43 negative, and the fatty acid chain. This PO43 negative is making this membrane, this cell membrane, a polar membrane. Now, what is happening is if we bring a electrical energy change near the phosphate head will try to deflect, especially if we are bringing a negative charge, the phosphate head will experience electrostatic repulsion and they will try to dislocate from their position. But the fatty acid chain will try to again accumulate them to their real positions, to their earlier positions, because of the hydrophobic effect. So the deflection of the phosphate head is the excitation time, which is polarizing the layer. The deflection of the fatty acid chain to again to their original position, the phosphate head, is called relaxation time. Is assumed as relaxation time. Now, using our nanorobots, we can generate a pulse which is greater than the excitation time, which is sorry, lower than the excitation time and greater than the relaxation time. This will result in a temporary opening of the nanopores on the cell membrane by opening volcanic shaped nanopores on the cell membrane. And these are temporary because the fatty acid chain will again bring these molecules back to their position and they will close the nanopores. This was this experiment was performed in vitro setup where we have stained the cell cytoplasm with a red dye and then the robots with the green dye, and in application while we apply a DC magnetic field or of particular amplitude, we can see that the robots are all present on the outside vicinity of the cell, whereas when we apply AC magnetic field of modulated amplitude and frequency, we can see that these nano structures are permeating inside the cell membrane and using the cell membrane as a scattering boundary. Again, the slides which were exposed, these cells which were exposed to the AC magnetic field with the nano robots, they were stack images of a confocal microscope, and you can see that in some of the slices, optical slices, these nano robots are present inside the cell membrane. But this was all you can say a stationary experiment. For dynamic experiment to see what is really happening, like in real time, we have fabricated this microfluidic structure, microvascular structure, microfluidic chambers, where you can see here in the mid we have replicate some of the human veins, microvascular structures, and try to do the experiment in this very minute space, which have a very controlled boundary. This was further analyzed using the SolidWorks design as well as using console simulations about the whole pressure gradient inside this. But the idea was we have to also attach the electromagnets at the two end of this PDMS blocked microvascular structure microfluidic chamber MSMC to expose the nanorobots to the magnetic field and perform its activity. Further, it was analyzed using bright field microscope or a fluorescent microscope on a bright field mode. And but before that, we stained the cell to see that how the cells are all like structured, or you can say assembled in this microfluidic device in this boundary condition. because here the nucleus of the cell is stained with blue, cytoplasm with red, and the robots are cell stained with green FITC dye and analyzed under the bright field microscope. Now here is the outcome of the experiment. First, I will show you the electroporation behavior of the nanorobot in particular magnetic field. So using this modulated magnetic field, we can see that the nanorobots can move from one cell to another cell, opening the nanopores, and then can get out of the system also. And a chunk of the material, which you can see a big chunk, cannot create this kind of behavior. And this nanorobot, because of this electrical field, fast electrical field changes, also exhibit intercellular signaling, because of which the central cell is rising up. One of the most important thing which we noticed during the experiment, so this was on one particular magnetic field where it can create the electroporation, but then it proves out that this nano vehicle is also the fastest cell targeting agent. So in another modulated magnetic field, which is now greater than the excitation time and smaller than the relaxation time, here the particles, these nanorobots will try uh, to not target any of the cells on its pathway. That means it will always avoid. Here we are trying to see the movement of the nanorobot, one nanorobot, through a cellular area, and you can see that no matter what, how many cells are there, what is the cellular density, it will always try to increase its speeds and go further and further. It will never stop. It is still going there, right from that position in the vascular structure. To further see this kind of activity, we perform another experiment where we have controlled the cellular um, density in the medium. Here we will try to see this movement of one nanorobot in the cellular medium, and here you can see that first it have a slow speed, but then more and more cells it will face on its pathway, untargeted cell, it will increase its speed, and then it can be taken to desired location, unlimited distances in the cellular medium in the controlled boundary condition with a very high speed, with an unassumable high speed, and such a high speed can never be seen in any of the nano vehicle or nano carrier or drug carrier yet proposed. You can see it's still going. Now it is increasing its speed because the cellular density is increasing. The slingshot mechanism, mm, where it is just because of electrostatic repulsion, it is further moving and moving in the excitation magnetic field. And when we stop the magnetic field, we can stop it to one particular area, to the targeted area, and perform the activities, whether electroporation or drug delivery or whatever we need. So in future, we believe that this vehicle can also be proven as one of the fastest vehicle in the cellular medium to perform these biomedical applications. So using one magnetic field, we can permeate inside the cells. Using another magnetic field, we can now avoid any of the cells on the pathway. So the key application of this technique is that in case of cancer cells and normal cells, the difference is cancer cells cell membranes Young's modulus is very different. Its Young's modulus is lower, and its cell membrane loses its integrity. Now at one magnetic field, what will happen is we will avoid the cells. At the same magnetic field, it will avoid the normal cells on the pathway, and at the same magnetic field, we will penetrate inside the carcinogenic cells. So such an experiment was performed here with scope with the ovarian cancer cells and healthy ovarian cells, and you can see that at one particular magnetic field and frequency, it was able to penetrate inside the scope 3, the carcinoma ovarian cells, and it was unable to go inside the healthy ovarian cells. So this proves that at this magnitude, at this particular magnetic field, we can have a targeted form of treatment of these ovarian cancer cells by by releasing the chemotherapeutic drug to those targeted areas only, rather than the chemotherapy technique where the whole body is exposed to the radiation because they were not able to distinguish between or particularly target the cancer cells rather than the healthy cells, and they kill all of that, which have a very high side effect. Next is, so this was all about the AC magnetic field. Now using the DC magnetic field, we can use these nano robots for one particular functionality, which is cell transport. So here you can see that the same microfluidic chamber, and we have the magnetic field, the DC magnetic field is switched on, and you will see that how these nano robots will push the cells going from high low pressure area to high pressure area in a very controlled form, and and uh, uh, fulfilling or completing the void areas. So how it is, it have a great application. I will show you in next slide. But here you can see that the cells can be transported by the robots slowly and slowly, and you can see the void area is completely now filled with the cells, and irrespective of the pressure gradient. In the next video also, I will show a higher magnification picture where you will see that how these robots are carrying the cells in an adhered cell areas. I will just go to the video a little bit faster so that we can finish it soon. Here you can see now how these cells are pushed over and slowly in this microfluidic chamber with variable pressure gradient, it is filling up all the void spaces in the pathway. Yes. So next video here is the higher magnification video where we have targeted some suspended cells using the robots, and then try to move these suspended cells using the magnetic field through a adhered cell area without disturbing any of the adhered cell area on the pathways. Here you can see that the robots are here on the cell membrane, and they are pushing the cells through these adhered cell areas in a very controlled fashion without any disturbance to the adhered cells areas. That means if we have nerves in the areas, so it will not disturb the cells of the nerves. Rather, it can pass over these suspended cells in a very controlled way. So, how we can apply this cellular transport technique by using the robots? The research have shown that when like patients with Alzheimer's or Parkinson diseases, they have the connection being break with the central nervous system or the brain, because the cells on the pathway loses their water molecules, which is the basic carrier of the electron path or the neuronal paths. But recent research shows that if fresh cells are flowed through these areas, through these narrow junctions, they will again try to regrowth the neuronal pathways. That means they can again reconnect the brain or central nervous system with these broken pathways. But we are talking about such a small minute scale areas that they cannot be reached out by any surgical technique. Our proposed device can only be the device which can be proven right now to perform such kind of cell transport, fresh cell transports to these broken pathways to regenerate the pathways. So using a DC magnetic field, as you can see here, how they are attached to the cell membrane by DC magnetic field, we can create techniques of Alzheimer treatment, vision reconstruction by corneal reconnection with the brain, and also reconnecting signalings with transplanted organs. On the second way, using the AC magnetic field, modulated AC magnetic field, we can target cancer cells in healthy cellular areas and deliver the chemotherapeutic drug by electroporation, or we can deliver foreign DNA molecules or foreign DNAs or biomolecules to targeted cells for for uh, for various techniques, for various if RNAs are delivered to these areas, they will create their own defense mechanism for further cancer spread itself. Lastly, here before using these robots, we need to also test their biocompatibility, which was tested with human epithelial cells and neuronal cell, rat neuronal cells, which are very sensitive to electrical field, and we see no cytotoxicity from until 50 microgram per milliliter, which is a very high concentration. Next is so since we can manipulate the surface charge of these nanorobots, first these nanorobots can be made more biocompatible by a porous silica coating, and then this porous silica coating have porous pockets on its surface. These porous surface can again be the surface charge can again be manipulated by negative increase or positive increase, as you have seen in the off-axis electron holography measurements. And then these pockets can be again filled with biomolecules. For example, here like chemotherapeutic drugs. Here was an example of the FITC molecule loading on these porous silica pockets using the magnetic field, which creates the surface charges. Protein molecules, which or some of the biomolecules which have the ends of say oxygen, that means two electron negative charge is there, so we can create the surface charge. We can modify the surface charge to more positivity and then attach these drugs, and then at the point of delivery we can release the drug again by giving a bipolar pulse, magnetic pulse. So this is the technique of loading and unloading of the biomolecules. This is one of the experiment where you can see this AGDTB drug, which is anti-HIV drug, can be loaded as well as unloaded from the nanoparticle, from this nanorobot, by using pulsed magnetic field, bipolar pulsed magnetic field. Lastly, as I said, we need to also deliver the DNAs for the mutating the cell or modifying the cell. So these DNAs can be loaded into these pockets and controllably released on the targeted sites. So in a gist, the whole experiment, the whole technique is that we have core shell, the biomolecules loaded, cancer cells and the normal cells. They will detect the cancer cell, avoiding the normal cell, open the nanopores on the cell membrane, go inside the cell membrane, and release the drugs. This was the main technique, which can be done by using these nanorobots. Further, this research was recognized by various news reports, various science agencies, as well as the Guinness World Record. And further, to control the exposure, like we cannot inject it in the bloodstream and let it go like that, right? We need to controllably release them in a particular targeted area. So for that, our current techniques is how we can capsulize them in these silica nanofibers, and then use a bunch of these nanofibers and then coat it with the pH sensitive resin, so that they can only dissolve in the particular targeted area, and then when they're in few microns distances from the targeted area, these nanorobots can be released, and then they will be manipulated by magnetic field and imaged using MRI. So such technique is not new. This MRI technique, with along with this Octamax for controlling the movement, is currently there in IIT Zurich, as well as some of the labs of magnetic speciality, where you can see that a micro robot can now be moved in a rat's eye, in a targeted site in a very controlled precision, in a micro scale controlled position. So that was all from my side right now, and these are the references which I want to quote, which have helped in my researches, in this research, and also support our idea. And thank you. And I was also told that there will be an honorarium for this presentation, which I want to donate to IIT's rapid action COVID, like for the rapid action COVID situations and for COVID patient reliefs. Thank you. Now I'm open for the questions.
Yeah, so thank you, Soutik, for this amazing lecture, very stimulating and insightful. And of course, very exciting to see your finally see your world record, discovery of these nanorobots. It's very exciting. So I request everyone to either type your questions or just send a private chat to me so that I can unmute you. So whichever is convenient to you. So the floor is now open to questions. Uh, yes. So. Yeah. So yes, so the first question which I have is from Siranjeevi Gurumani. So the question is, what about the time span of bioavailability of the nanoparticles?
Okay, time span of the viability of nanoparticles.
Yes.
That's the question. Okay, so
Yes.
viability. Okay. Maybe if I don't understood it correct, but viability, that means like exposure time of these particles with the cells. That's what is the question.
Yeah, I think the question means something like that, like probably how much time it actually gets exposed to the cells, and probably at what
Yes.
concentrations, you
Yes,
know. Yeah.
that's a very important question actually. So if you see that if we are looking into the cells which are untargeted, which are not targeted the pathways of these nanorobots, they are really not staying even microseconds to these regions. Whereas when we are trying to create an electroporation on a targeted cell, we need to spend few of the milliseconds. When we are delivering the drug at a particular site, we also again need this millisecond time scale, because we need to change the magnetic field quickly. But with this exposure, the exposure of these cells, these particles at the to the to the to the human body or to a living body is much less than the dust we inhale every second. That means that in each exposure of these capsules, for each treatment exposure, the amount is much lesser than the dust or foreign particles we inhale every minute or every second.
Okay. So also another question is, how long these nanoparticles exist in the system? For example, inside the body, like how much would you expect it to be hanging
Yes,
around? Yeah.
this was the question I always got from various places, because there are many nanoscale devices which can be used for drug delivery, but the disadvantages were that they stay in the system. There was no technique to take them out back. But here we can take them out within a millisecond time scale, because of this, as you saw in the first video, how it is moving from one cell to another cell to another cell. And since they can penetrate through the smallest unit of our body, which is a cell, they can be taken out right away from the skin by sweat, rather than from the urinary system, or from any other system where there is a chance of accumulation.
Okay. So the next question is from Vishnu Arumugam. So what are the challenges in characterizing the locomotion of these nanostructures? If we go even below 50 nanometers, is there any techniques to follow the locomotion?
Yes, actually, the thing is, right now, since I have shown you the in vitro tests where we were using particularly bright field microscope, and we are only analyzing through one small nerve. But in case of in vivo experiment, we have current technique called NMRI. Since there will be a group of these nanorobots which are exposed to the cells, these can be particularly tracked, and also they are only exposed to the cells coming out of the capsule when they are micrometer precision distance, when they are few micrometers far away from the targeted area. So when we will focus our NMRI to those areas where we need to release the drug, it will not be very difficult until this 50 nanometers scale, but it will be difficult if we go further below, particularly because then the loading itself will be very difficult. The loading itself will be very difficult because controlling a 15 and smaller than 15 nanometer using optical microscope, capsulate them itself will be a little bit difficult, but not impossible. There are techniques which also shows how we can insert them inside the nanotubes in aligned nanotubes by the capillary action method. Yes.
Okay, okay. So there are a few more questions. So I think the next question is pretty interesting. So Manoj Kumar Sharma, his question is, can we use it to deliver DNA molecules inside the nucleus of the cell by modifications? Because we only talked about cytosol delivery. So is it possible to deliver molecules inside the nucleus?
Yes, it is possible to deliver the inside the nucleus, but it have both the positive and the negative side. Yes, we for the DNA we definitely need to deliver it inside the nucleus. But if we don't have a controlled movement inside the cell, then it will create it can create mutation in any of the cell where we are going inside. But yes, it can be used for delivering the DNAs inside the nucleus of the cell.
Okay. So Gokul asked, like, when can we expect these nanorobots to be commercialized?
Yes, that's what is the question by Czech Academy of Science, also from AIMS, that when we can expect the commercialization. Right now, there are further things which need to be done before the human testing. But yes, currently we have some of the devices for Medtronics where we can perform kind of cancer cell detection. Earlier, we need to do the biopsies, send it to labs, and then it will take 2 or 2.5 months to really get the results. But this is an instantaneous result by culturing those cells. Secondly, for the precision medicine, for testing the precision medicine on targeted areas, these in vitro setups are currently being proposed and also in the way of patenting to be commercialized. But for the ultimate outcome of this research will need more 3 to 4 years to really come up and take out the chemotherapies for first two stages of cancer. The third stage will be much more exposed, so nanorobots will not be able to do that for such a big exposure. But for first two stages of cancer, they will definitely be used.
Okay. So hopefully it happens soon. And also, like, uh, so another question is from Shomik Rachaudhuri, but I think it is already answered. Like, yes, it can cause some mutation or change in genetic information if you can put molecules inside the nucleus. Uh, so, uh, so another question is, since nuclear intake is not properly controlled, is there a guarantee that the particles will not enter the nucleus or even go near its vicinity? So, yeah.
Well, yes, this is one of the research, one of the publications shown by some of the researchers that how much time you need to really change spend in the nucleus vicinity to change the to create the mutation, because sometimes the mutation is created by electrical deformations, sometimes by chemical deformations. So the idea is if we since we can use it for DNA transfer inside the nucleus, in that way we need to have a higher exposure time. But if we're trying to avoid the nucleus, in that case, this millisecond will definitely not create the chemical changes because the diffusion time is much more than the time it moves to 10 micrometer cellular distances.
Okay. So I think you have answered almost all the questions here. So I think one of the last questions we can take is, what about protein structure modifications due to the electric field in the microenvironment? So,
Yes, uh, this is one of the thing which I already mentioned, that in case of precision medicines, where earlier it was a hit and trial method, various biomolecules or protein molecules or like sequencing RNAs are like were inserted and try to check that whether they are creating the defense mechanism or they were created they were there to create the molecular integrity of the cell membrane due to protein deformation. Yes, this it is already being analyzed, and there are some of our papers where you can read about this work that how they can be used for this modification in a targeted modification in vitro setup.
Okay. I think we have come to the end of the Q&A session, but finally, I would like to ask a short question. Is like, what was the previous record, and what was the record you broke for this Guinness Book of, Guinness World Record? So
Yes,
was
actually,
there
actually, before this, I was looking into the system. A record by ETS Zurich
Mhm.
for a micro scale machine, which was used for cardiovascular detections, like hole detections, and also like cardio artery clearance, and those dimensions were almost like 700 micrometers. So we have broke this record by almost 1,000 times smaller scale.
Okay. Yeah, so that's wonderful to hear, and we expect and we hope that many such nanorobots come out of your lab from IIT Delhi and solve a lot of biomedical problems around the world. So all the best, Dr. Soutik, and thank you again for accepting our request and sharing your research with us. And particularly in this pandemic times, when we are trying to kickstart our research activities, I think it will provide a lot of motivation to our young students and all who have attended the lecture. Thanks again, Dr. Soutik, and I think we will have you sometime physically in our department when the things normalize again. Thank you.
Thank you very much. And as you can see that we need a very big collaboration with chemical and biomedical and electrical together to come up to bring this technology in 4 years for human welfare as well.
Sure, sure. Looking forward to it, Dr. Soutik.
Thank you.
And before we leave, I would like to thank Professor Anil Elias, our HOD, and also Professor Ram Ramaswamy for their constant encouragement, and also Professor Janakiram Vaitla and Professor Vishwarup Chakravarti for co-organizing this webinar series along with me. And on behalf of our department, we would first of all like to thank all the attendees for attending the webinar today, and also we'd like to wish you good health in these difficult times. Please take necessary precautions and stay safe. And hope to see you all soon. Have a great evening, everyone.
Same to you.
Okay.